So it's my pleasure today to introduce Dr. Georgiadis. He did his bachelor's and master's in biomedical and mechanical engineering in Greece, and then he moved to ETH in Zurich, where he did a master's in biomedical engineering and a PhD in bone biomechanics, where he started developing the techniques that we'll talk to us about today for imaging bone. Then he started his postdoc in Zurich, applying these, translating these techniques to image fibers. And then he moved on to NYU, where he used this approach to validate diffusion MRI. And now he's at Stanford, where he's applying these techniques to study degeneration in Alzheimer's disease. So please join me in welcoming Myos to the subject.
Thank you very much, Anastasia. It's my great pleasure to be here and be able to present this work, which so it's titled, it would be Quantifying Myelin Levels and Axonal Orientations using X-rays and some correlations to MRI. Unfortunately, not validated diffusion MRI yet, but yeah, that's one of the goals. And I will start actually by some acknowledgements, as Anastasia mentioned already. This is the work present here was started in ETH Zurich, and then moved to NYU, and recently, actually quite recently, to Stanford. So a small part of this will be there. And most of the work, so I will be talking about X-rays, but not the usual X-rays in CTs, but stronger X-rays which need synchrotrons. And there are some places, or a few places around the world, where one can find these like huge particle accelerators which produce very strong X-ray beams. And so most of the work has been done in the Swiss light source in Switzerland and in the Brookhaven National Laboratory in New York. It's actually in Long Island. And fortunately, there's also one next to Stanford, the SLAC National Accelerator Laboratory, where I will be doing work. And most of this work has been funded by the Swiss National Science Foundation. So, starting from a really broad perspective, so we have nice tools nowadays to image tomographically tissue, and we have samples can be like at the meter, let's say, scale, but we have also higher resolution techniques where we can go to the centimeter, millimeter, so micron scale, even nanometer, and it is all these scales that have very interesting properties for our large samples also. So ideally, we would like to be able to translate properties from the nanoscale to the more broad scales. And today I will try to show how this can be, to some extent, approached with using X-rays, because luckily X-rays can go through a matter, and so we can get deep in tissues. So that's a spectrum where we can see here the soft and hard X-rays, and there's different techniques which are applied depending on the energy one chooses, and usually interactions of X-rays with matter involved mostly, of course, there's a transmission, so X-rays, that photons that just go through, but also part of them gets absorbed or scattered. And here I want to highlight the main phenomena. So one of them is like Compton scattering, which is inelastic, where a photon displaces an outer shell electron, and which can produce also radiation damage. And also a lower energy photon is the outcome of this. There's the photo effect, where an inner shell electron is displaced, which also can also cause radiation damage. And also when it's replaced, we have characteristic radiation. And the effect, which will we'll be most talking about today is elastic scattering, where the photon goes and practically keeps the same energy but changes direction. So that's why, hence the elastic part. And so what happens is, in the end, we have, so it's scattered at a specific angle, and we have the so-called momentum transfer, which tries to quantify exactly this. It's kind of this angle of scattering. And this is not, so most photons don't go through this process. So this is the equation linking the incoming beam, the I naught, with the scattered beam. And especially this term here is really small. It's like something like 10 to the minus 30. So the probability is really low, but fortunately, this is the probability from one electron, but fortunately we have many electrons. So 1 milligram of water has 10 to the 20 electrons. And also if we have many photons, we can have nice statistics. So how it works, if we zoom out a bit, X-rays hit a sample, and part of them just goes through, which is the transmitted beam, the primary beam, but part gets scattered. And actually in CT, we usually filter out this scattering because it would blur the image. So we have exactly anti-scatter grids. But these scattered photons contain really interesting information, and information becomes very interesting where we have an organized microstructure, especially repetitive one. So we have constructive interference of the photon waves. So if we place a detector downstream, we can record really nice so-called diffraction patterns. So to the left, you see some typical crystallography images because of the crystal lattices. One can also see effects like edge scattering, where it's not due to a repetitive microstructure, but just due to edges. And also from organized macromolecules. One, I don't know if this image looks familiar, but it's the one on which the Watson and Crick assumption of the double-stranded DNA double-stranded helix was based back then, when we didn't have electron microscopes. So it was one of the first ways to really get nanostructure information. So now we can take that and apply it to different tissues. There is a lot of tissues that this can have applications on. One of the most abundant proteins of the body, collagen, has really nice repetitive structure. Also muscles have the actin-myosin filaments that are repetitive, and we can get information about, for instance, relaxation and contraction. Today I'll be talking mostly about brain tissue, where we have a really nice repetitive structure, which is myelin, the myelin sheath, which effectively X-ray photons, when they hit and the myelinated axons, and they see this repetitive structure. So downstream, they produce, at the detector level, they produce these like orange or yellow, these rings, which are the myelin rings. And this is how it looks in a real life detector. And actually these black lines are just due to detector electronics. But these rings you can see are specific to myelin. And actually the distance from the center corresponds exactly to the repeat distance of myelin. So we can probe the nanostructure. As the X-ray goes through the sample, it probes the nanostructure, it probes myelin, and gives downstream these myelin rings. And this is again the same Q is the reciprocal space, is the same momentum transfer that I mentioned before, which is actually the angle of scattering, which is linked exactly to the repeat period. So based on this information, one can get myelin content information, because if there is any myelin, it is going to scatter in that angle, and we are going to see these myelin rings. And one can get also neuronal directions based on that signal, because you see that the axons here are upright, and they have So the photons get scattered right and left. But for instance, if they were just horizontal, the rings would be in the perpendicular direction. So would scatter kind of up, down.
So if you would have like the same but covered in the whole,
Sorry?
if you have the same, maybe like the axons horizontally, but they are like covered in maybe the whole view that you have, you will still detect that they are horizontal?
Yes, yes, because this, so the beam that goes through probes the 17 nanometer, so repeat distance. So as long as the beam is larger than 17 nanometers, so that it can probe, you can have this coherent, so these constructive scattering effects, you would see these myelin rings. So you would see them if you have like a small sample, a big sample, you would see them. So I'll first start talking about neuronal orientations, and actually people not so how you can get directionality information, and people have done that actually since quite a long time. Looked at two-dimensional patterns of orientation. So here you see different tissues. That's bone, teeth. That's I think pig heart or liver, I'm not sure. But so the point is that you can get these color-coded maps which reflect 2D orientation. So that was the status kind of when I started my PhD. And as mentioned, I worked first on bones, and I would have bone sections where I would cut the bones in thin sections and then go to synchrotron. I would raster scan the sample, but also rotate it. So by rotating, you can get information how the signal changes with rotation. So you can kind of, by fitting this signal to an equation, you can get a three-dimensional orientation of the fibers. In that case, it's the mineralized collagen fibers in bone. And if you just stack the consecutive sections together, you can get information about the volume. But of course, ideally, we don't want to section the tissue. So we came up later with a tomographic method to do that, where we would raster scan the sample and also rotate it and get information from multiple angles, multiple projections, kind of similar to CT. But in that case, we didn't reconstruct just the density value as in CT, but we would reconstruct multiple parameters per voxel, which is a quite more, let's say, complicated problem than reconstructed as a single value per voxel. So we name this technique small angle X-ray scattering, because this is the photon scattered at small angles, and tensor tomography. So now, taking that to brain tissue, of course, we also have diffusion MRI, which can look at fiber orientations. So starting the experiments, first MRI, could be both in vivo or ex vivo. Take the very same sample, sectioned it with a vibrotome, got sections, and then did similar experiments to the bone sections, where we would raster scan and rotate. But first, even without raster scanning, so this is just 2D information for a mouse brain section, and you see, for instance, how scattering from different parts of the brain look like. So first of all, this is color-coded according to these 2D orientations. So whatever is horizontal is red, and for instance here, and so on. And you see that so from outside the brain, you don't have practically almost any signal. And then from different, when we go look at white matter regions, you have these myelin rings, and their orientation depends on the orientation of the fibers. And the gray matter, you have very little, if any, except from the molecular layer, the outermost layer here, where you have a bit more actually myelin here. So this, correlating this with diffusion MRI, here is 2D information only, which looks kind of similar. And of course, we want to have this in 3D. So one could get information, as I mentioned before, just like vectors, but also that recently came out, how we treat it so that we can have a orientation distribution function, not just vectors representing the fiber orientation, so that we can get for instance, here with a tensor representing the orientation distribution function. And looking at part of a section, this is the 3D orientation with the typical color coding here in 3D from X-ray scattering, and also doing diffusion MRI, finding the same part. Now, here I have mirrored it, and seeing that this looks like very similar orientations. So when trying to quantify it, the dot product, so again, the main fiber orientation, it's the dot product of the two vectors is really skewed towards one, which would mean that they are the same direction. Now, the degree of anisotropy, because in diffusion MRI we use FA, sorry for the, hope the letters are not very small there. But this is FA from X-ray scattering on the x-axis and from diffusion MRI on the y-axis. And you can see that there is not like a perfect or R equals to 1, because the correlation is not perfect, because it's very different mechanisms that give us this degree of anisotropy. And yes?
So did you downsample the X-ray data to the resolution of diffusion, or did you acquire it at the same time?
So this actually, yeah, so this I tried to kind of match it in that specific case. So the left is with 50 microns, and diffusion was 75 microns.
So how was the brain repair? Was it fixed in therapy?
Yeah, so in that case, it was fixed formal. So the same, I took the same brain that was scanned with a diffusion MRI ex vivo, and in that case, it's sectioned and with a vibratome. Nothing, and put in between.
There is still water in?
Yes. So I had it. I had it here. So actually, this here, it is in a kind of water chamber. So it is in between. This is, I think, in that case, 500 microns of water in a water chamber. And this is just the section which has stuck to the one side. And I have put it in a water chamber, like PBS. And so, of course, you can see other, like some geometrical distortions here. For instance, ventricles are larger, which is due to sectioning. And of course, we don't want to have that. That's why we have the tomographic method. So doing experiments on the tomographic method, taking the whole now brain, putting it in a tube, again in PBS, usually in gel, agarose gel, and scanning. And so for instance, I have here the brain, and here the diffraction pattern from this beam, which is here in red. And this is a video as this beam moves through the sample. I don't know if you can see it, this red beam, you can see it when it gets in the sample now. So you see the scattering changing. This is in gray matter still. And when it reaches white matter regions, we can see hopefully these myelin peaks there, these rings at the diffraction patterns. And the idea is, this is done in reality faster. This is just for display. And the idea is that you go and raster scan the sample and then rotate it, raster scan it again. So you have multiple, this information for multiple projections. For each projection, one can get such a 2D orientation map. This is again information in 2D. And you do that for all projections. So here I have the different projections, so rotating the sample and scanning it. And for reference, I also have the absorption image, X-ray absorption, which you see almost nothing there. But scattering can give you really nice information, and myelin specific. And then one can do the so-called tensor, X-ray scattering tensor tomography, which the initial method was based on spherical harmonics. And then realized when starting doing experiments in the brain that it's a really complicated structure in bone. Fibers wouldn't change directions as abruptly. So we kind of stepped back in complexity and did another method which was based on simple rank 2 tensors, so kind of ellipsoids, similar to DTI. And this method, so we'll demonstrate this in carbon fibers or bone trabeculae again, but also on the mouse brain. And this is now a tomographic virtual section through the brain where you can see the fibers, the fiber orientations represented by ellipsoids. And here I also have some very basic tractography based on that. But so this is looking tractography, just plain looking at the main fiber direction. But this is from myelinated axons only, which means, because ideally, so for tracts, we want to, it's the myelinated axons that transfer the information, so the signals, long range. So ideally, this is what we would like to have, information about fibers which are the myelinated axons for tractography. And going further from a mouse brain, actually doing more metrics, for instance, comparisons with diffusion MRI, and here I have with the ROT-in framework, the rotational invariant framework, which from the NYU lab. So here I have a correlation between the P2 anisotropy parameter from the X-ray scattering and diffusion MRI, the ROT-in framework, showing these are the ROIs looked, showing really high correlation. So one can really start now really correlating the fiber orientations and from X-ray scattering, which are specific to myelinated axons, to ones from diffusion MRI. And going further from mouse brains, for instance, in spinal cords, which also were giving really nice scattering signal. Here I have two projections, one from the front and one from the side. And then one can tomographically reconstruct this tensor based on X-ray scattering, and again do, for instance, comparison between fractional anisotropy or the P2 anisotropy parameter that I mentioned here, showing that the actual P2 captures anisotropy better, because FA also has the isotropic part in it, which is method dependent, whereas ideally we want to quantify anisotropy, which is with something not related to the underlying physics. And also can do tractography, this is more like probabilistic tractography here, and showing nicely, for instance, sensory incoming tracts in the dorsal horns, and also the motor tracts going out in the anterior horns. And this typically is nicer than, I also have diffusion tractography for this, and these are not quite as nice. And the idea is that because there is, for instance, these tracts, the sensory, and in the horns, there is a lot of cells in there. So diffusion struggles to get really nice orientation information. Whereas here, we can be specific to the myelinated fibers and pick up the few myelinated fibers that might be there within a large cell population, and get nicer tractography due to that. Also now going to human samples, this is an example from white mat, so from the corpus callosum body, and doing again diffusion. Here that tractography is not that interesting because it was quite homogeneously oriented. Fibers were quite homogeneously oriented, but still the idea is that it could be done, it can be done also on human specimens. Now I'm going to switch a bit and talk about myelin content. And here, what I already mentioned is that from a single point, you get this diffraction pattern and these peaks that correspond to myelin. And one can isolate the myelin specific signal. And one can do that for the whole brain and all projections, and then tomographically reconstruct and like that, one can get a myelin map. Here's a 3D map, and then virtual slices through a 2D map. And in MRI, there are multiple methods which are myelin sensitive. So what I did is I had scanned the same brain with, here what you don't see is magnetization transfer saturation. So the two top techniques are based on magnetization transfer. Here's T1 and T2 maps, and these are diffusion, different diffusion metrics, fractional anisotropy, axonal water fraction from the white matter tract integrity metrics, and this radial diffusivity, and this is extra axonal perpendicular diffusivity. And ideally, we would like to have also a gold standard for this, some ground truth. So I took half of this brain and did the myelin histology and extracted the myelin, isolated the myelin signal. So this was half the brain, 320 consecutive sections stained with Luxol fast blue. And then taking this half brain registered to the X-ray scattering and MRI data set and did correlations over all the voxels to really try to have correlations with myelin metrics. So here you can see the different myelin sensitive metrics, and the first two rows are correlations with histology. This is a Pearson and this is a Spearman correlation, and you see that as kind of expected, because X-ray scattering is specific to myelin, this information, so it has the highest correlation, so linear, but also monotonic correlation with histology, and the best MRI-based myelin content measures were based the magnetization transfer related ones. And then the idea is, what I want to propose is that this can be used as really a quantitative method for myelin content instead of histology, which has introduces actually many artifacts in there, and it's really hard to quantify and hard to get volumes of it. So this, you can take the exact same sample and do tomography with the X-ray scattering tomography, and then you can even take it and do whatever else you want. Because exactly it's a tomographic method and also myelin specific method.
And is that MRI ex vivo?
Yes, that's ex vivo.
That's why the T1 has been published.
Yes, that's a good point, yes. And so in the third row here, I have the same correlations as with histology in the same row, in the second row, but now correlations of the same metrics, the MRI metrics, with X-ray scattering, because the idea, what I want to suggest is that one can use X-ray scattering as a reference method for measuring myelin. And as you can see, correlations are very similar to the ones with histology. So again, second row correlations with histology, and third correlations with X-ray scattering. And now taking these correlation coefficients, you can also see that in a, they have a really, these are very highly correlated, and also in a, similarly in a Bland-Altman analysis. Sorry. Here you can see that actually the coefficients are almost the same, with a reproducibility coefficient of 89%. So, which means that they practically measure the same thing. So now going to illustrate a bit better the nanostructural sensitivity. So one can really, because it's, X-ray scattering is sensitive to the myelin periodicity, actually central and peripheral nervous system have different myelins, types of myelin. So in the central nervous system, there are the oligodendrocytes, which build the myelin around the axons, and there the repeat distance is around 16 nanometers, like 15 to 17. But in the peripheral, with the Schwann cells, these have a periodicity of 20 nanometers. So these give actually distinct X-ray scattering signals, and so you can really distinguish the signals from the peripheral and the central nervous system based on the periodicity, because remember, the distance from the center really tells us the periodicity of the structure. And so one can treat the CNS and PNS myelin separately, and then what we are looking here is actually mostly this green part here is the trigeminal nerve coming out of at the bottom of the brain. And if one does tomography on that, one can see really nicely, one can reconstruct separately the central and the peripheral nervous system. And one can even see a really nice fit here. It's a kind of ball socket fit in the trigeminal nucleus, where the central nervous system myelin stops and the peripheral starts. And yeah, one can do the 3D rendering of a combined. So here's again the mouse brain with a trigeminal nerve sprouting out. So finally, I want to talk about the possibility to study dysmyelinated models, so myelination diseases. Here I selected dysmyelinated model, the shiverer model, which actually the myelin does only a few wraps around the axons, and these mice die very young. They're called shiver because they tremble, they shiver, and they die very young. And compared to the compact myelin, which you can see here, has really many layers. So we tried myelinated control, shiver, and they treated mouse based on a glial progenitor. So it was an injection with glial progenitor cells. And here is kind of a video how they took, like in the beamline, how it looks like the scanning. And so here, this is the mouse. This is the overall scattering intensity, not myelin specific, which is actually this curve, this blue curve. And what one can do, one can isolate, again, as mentioned before, the myelin signal only. So you can see as this, you can see maybe, I hope, the beam going up through the sample. And when there is myelin, you can see the peaks getting specifically isolated. So one can get myelin specific information. And in some parts, there was also some peripheral, so you would see some extra peaks. But the idea is scanning again the whole mouse. With that, one can get myelin information from that, and doing that for different projections. So that was the control brain. And also this is the successfully treated, where it has, we knew that from this treatment, it would induce periventricular myelination in theory, but we would like to quantify that and any kind of treatment, the success of the myelination. And you can see here that mostly there is nothing. There's no myelin peak. Apart from there are some small peaks at the central area. And actually this is what we saw for both these. I have the example of these two projections here. You see only periventricular myelination. And so we can really localize the myelin in a tomographic way. And here I have the comparison to the shiverer, where you see that these, so shiverer mice have these like very few layers of myelin around the axons, like 2-3 layers, but this could still be picked up here with X-ray scattering. But you see scales are also quite different. So this is 0 to 1. So this, the amount of myelin here is really much, much less than the normal myelinated control. And here I have the comparison of the 3 samples. So the RAG2 is the myelinated control, TX is the treated, and shiverer here for different ROIs of the brain. So this Y is myelin levels, and this is different ROIs. So here the genu, body, and splenium, corpus callosum, then internal capsule, thalamus, and cortex. And you see that the myelinated control has, as expected, in the white matter regions, high myelin content and low in the cortex, lower, but there is still there is myelin there. The treated mouse had more like towards the genu, which could get to almost the levels of the normal myelinated mouse, but then it would really have almost very little myelin in other regions. And you can see that the shiverer has some really little amount of myelin, but there is still some myelin overall. But even in the white matter regions, it's lower than the normal cortex, myelinated mouse cortex. And here, trying to visualize that, I have, this is a kind of video showing So I start, it's a kind of moving threshold. So I'm starting on myelin level 12, which is up here. You don't see any myelin, and I will start going further down. So in theory, you should start seeing myelin here first in the myelinated control, and then in the treated here, just in some regions, and shiverer really low. So this, I hope that plays. Yes, so you start seeing myelin levels where it's highly myelinated in the control, and at some point you start seeing the myelinated regions, and you see that this is already, you see all that myelination in the control, and it's then where the shiverer myelination starts showing up. So it's really when, it's a pity that this now looks like that, but yeah. Anyways, so this here, you could see, what you could see is that it's almost everywhere myelinated here until you get to the levels that of the shiverer myelin. And you can really quantify region specifically how much myelin is in the treated brain. So that was the overview of what this method can do. To conclude, so MRI is really a nice tomographic method which can probe microstructure with different methods, diffusion MRI or not, but we need structural validation. And X-ray scattering is very sensitive to organized nanostructure, and in the brain tissue can provide myelin content and fiber orientation specific for myelinated axons. And actually you can take the exact same sample. So it's just a fixed sample, and you take it and do tomography without any need for any extra sample preparation, and then you can do whatever else you want with the sample. And the idea now is to go in the study of human neurodegenerative diseases and try to investigate the possibility of in vivo scans first in rodents. And actually, this is why I have been in the East Coast in the past days. I was doing experiments on Alzheimer's specimens in the NSLS, so the National Synchrotron Light Source, which is in Brookhaven in Long Island, close to New York, not very far from here. Actually, I think it's 3 hours drive or something. And here is the beam line. So the sample probably you can't. So it's here. And so zooming in, it's this guy where you have all these fans. So X-ray beam comes from here, and downstream here, the photons, the scattered photons, go towards the detector. And actually, I could see really nice scattering from these samples too. But haven't had time to analyze it yet. So maybe next time I can show some results for Alzheimer's specimens. Yep. So I would like to thank you very much for your attention. We will be glad to answer any questions.